zetten.
So hello everyone. It's really funny and fun to see everyone pop up on my screen every time. My name is WdKa Groenveld. I'm an advisor from the beginning at the Science Gallery Rotterdam, where we try to improve the dialogue between science, art, and young people, bringing together scientists, artists, and young people, and get them to talk to each other, to see the different ways of looking at things and learn from each other. It's already the fourth inspiration session, the last one of this Studium Generale, together with the Willem de Koning Academie Lifestyle and Transformation Design. We have organized this with all kind of people, artists and scientists from the Erasmus MC. This week we already talked about the pandemic, zoonosis, basically where does this COVID come from, but also the indirect aspects of COVID, also socially, physically, mentally. Trust in science was the third one yesterday, and today we will be talking about future perspectives. We will be talking about that with Luc Coffin, who will guide us through how to build models, like his model for group immunity. After that, we will go to André Uiterlinde, who will get us an insight in how host genetics, so why and how does COVID impact a person more or less, and what kind of DNA kind of genetic background is back to that. And then there's Agnieszka. She with biofriction. She will also tell us about, will take us through different ways of looking at how, more futuristic aerosol knots, a futuristic tale. I'm really looking forward to that too. So without further ado, I would like to give the floor to Luc.
Thank you, WdKa. Good afternoon, everybody. My name is Luc Coffin. I'm an assistant professor at Erasmus MC. I work in the Department of Public Health as a full-time researcher in infectious disease control. And I was asked today to give you a little peek into what kind of work I do, basically. So I'm an infectious disease modeler, and I have a few slides to share with you. So bear with me while I share the screen. There we go. So infectious disease modeling, a short introduction. So, well, why are we using models? I'm sure some of you can relate to the question given the current situation. But let's think about an example. You can probably imagine something about an emerging new infectious disease. Suppose a new deadly infection has emerged and we know very little about it, but we want to control it as quickly as we can. Of course, we can do all sorts of field studies and experiments to answer this question, but by the time we find out, it may be too late. And even if we perform such studies, the findings of such studies may only be relevant for particular settings. So what we as mathematical modelers or infectious disease modelers do is, parallel to field studies and experiments, we perform simulation studies, where we build basically mathematical representations of reality, which are simplifications, and we use them to simulate and predict future events, much like the weather forecasts are produced. So an example, of course, is COVID-19, which is caused by the SARS coronavirus type 2. And as WdKa already mentioned, last year, I, together with a colleague, developed a mathematical model for this, along with a lot of other people. And this mathematical model that we developed was a representation of a country, its regions, its cities, and its inhabitants and their contacts. And on the right here, I'm showing you a graphical representation where each little circle represents a community, with the size of the circle representing how many people live there. And these circles are organized in little blobs that you can take to represent, say, neighborhoods or cities or regions. And this is an animation, so I'm going to start this for you now. The infection started in a few of these regions and starts spreading at some point until we become aware of it and decide to lock down the country, which makes the epidemic cool down, which is represented by the blue color here. But maybe then at some point we think, well, we're safe now, we're going to release measures, and then the infection explodes exponentially and the whole country is overrun with the epidemic. Now, this is a hypothetical scenario. Of course, because this is not what happened in reality. We're still very much in the stage now where most of the people in the Netherlands have not yet had corona. But the nice thing about this model is that it gave us a few important insights. First of all, that achieving herd immunity against COVID-19, meaning that enough people have already had the infection such that we will not have another outbreak. Achieving this state without a vaccine will take in the order of years. So that would mean years on end of lockdown, which is not a nice prospect. And there is no single intervention that keeps this infection in check. We really need a package, a combination of interventions. So how do these models work? Well, let's start with the basics of the basics. Almost a century ago, Kermack and McKendrick developed the very first infectious disease model, which is called the SIR model, where the letters stand for the 3 states that people are considered to be possibly in. Susceptible, presumably, and then maybe at some point during their life they can be infected, and after infection they may recover. So imagine that we conceptualize this population basically as these 3 compartments, and people can move between these compartments, much like, for instance, water flowing through tubes between 2 barrels. This is just a metaphor, where the speed of the water flow between the susceptible and the infected barrel depends on how many people were already infected in the first place. Because you can imagine, if there are more people infected in a population, more susceptible people will become newly infected. On the other hand, the speed at which the water flows from infected to recovered is a fixed speed, because that is basically determined by how quickly people recover once they are infected. Now, with this kind of model, we can explain the trends over time in the number of cases in each of these compartments. And these models have helped us understand why epidemics grow exponentially at first, but then slow down, which basically happens because you run out of susceptible people at some point. And then in the end, at the peak, there are just as many people getting infected as recovering. And then in the end, there's always a long, long tail. Now, this is a very basic model, and it doesn't really represent realistically any disease. So for real disease applications, we typically develop more sophisticated models, variations on this SIR model. For instance, SEIR, where we add a compartment that is representing the exposed people who have contracted the infection but are not yet infectious. Or we may consider a model where we stick an S to the end. So SEIRS, where people lose recovery at some point and become susceptible again, going back to the first barrel at the top. So we can think of many, many different types of models to describe any potential disease with this kind of structure. And for COVID, we typically use the SEIR or SEIRS model. And I just put some biological features of the disease on the screen. For instance, the latency time, so the duration between becoming infected and becoming infectious in COVID is 5 days, whereas the incubation time, which is the time between getting infected and getting symptoms, is a little bit longer, 5 to 6 days. So these are like the basics of mathematical modeling, and these have been used and developed for almost a century now. But we're now arriving at a very interesting frontier these days. And that is the fact that we are realizing more and more how infectious diseases interact with our own health-related beliefs and behaviors. Because of course, behavior determines the spread of infectious diseases. But these infectious diseases also affect our thoughts about those diseases, because if an infectious disease is spreading rapidly and we see about that and read about that in the news, we are going to adapt our behavior and our beliefs for the better or for the worse. I mean, we can discuss a lot about vaccination and anti-vaxxers and all that. So there is a continuous influence between behavior, beliefs, and infectious disease spread. And this complex interaction, this complex dynamic is something that we now try to better capture in newer types of models where we simulate the individual life histories of individuals in the population and how they interact with each other in social networks. And this is a blossoming field of work, especially inspired by the current corona pandemic. With that, I finish. I hope I provided a little view on the type of work that I do, and I'm happy to take any questions. Thank you.
Well, thank you very much, Luc. You can, of course, everyone can put a question in the chat in between. We'd like to put the questions at the end.
Okay.
What I think is really interesting is that you're putting beliefs and behavior into your models. That's quite a challenge too, I think. Let's go now to André Uiterlinde about COVID-19 host genetics and how the differences, how the personalized yeah, I'm sorry, how everyone has his own way of getting ill and etc. So please go ahead.
Well, thank you. Well, given the complexity of the topic, I suggested my title, Complex Genetics. Yeah. It's in Dutch, by the way, I realize now, but my slides are in English, so no problem. I took the liberty of taking this topic a little bit broader than just COVID, host genetics, although it is an important part, because I think most of us are a little bit bored with COVID now. We want to get rid of it, of course. But you heard from Luc how important the research on that is also for the phase after having, let's say, played victory over COVID. It's not over yet. And that illustrates what we as scientists are busy with. We are searching for many different things, most of all the truth, of course, what's happening. Then we try to decipher the things that we measure in people, for example, how to understand it. And then last but not least, we try to implement that in healthcare. Now, in relation to COVID, most of this you have been hearing about the implementation phase. So what's happening in the healthcare setting? There are not enough IC beds. The vaccine is being distributed slowly. Is there a vaccine? Yes or no? Do we have side effects? But all of this is preceded by a lot of research, like Luc just showed to you how we can model how infections spread. My talk is about how more in-depth knowledge about why people differ from each other can also help maybe in the end to help prevent a lot of things. I give here two Greek figures, Hippocrates and Pandora, to illustrate there are usually two sides to a event or a discovery or an implementation trajectory, both a good side and a maybe somewhat worse side. We have to think about and most importantly discuss with each other if we want this, yes or no. And I think at the end I'll come back to that. Let me see if this works. Yeah. Of course, you're all familiar with the fantastic diversity that we have as a species. These are all humans, as you see them, historic and still living. They're all different from each other. Most, of course, what you will see here is their behavior, like what Luc was referring to, but also in terms of what kind of diseases they develop and what kind of characteristics they have, the way they look, for example. And what we've learned over the past 50 years or so is that DNA plays a very important role in there. And then in particular, the differences in our DNA between the human individuals. We know now that DNA variation is involved in how people develop, how babies develop, how we look like, the appearance. Our behavior is also to some extent under genetic control, as we say it. The aging process as such, which diseases do we develop? Do people age early and quicker than others who age slower and live longer? And of course, I'm working in a hospital, the Erasmus Medical Center. We're very interested in, can DNA variation help us in understanding disease and maybe predicting disease? And this all together has led to this concept, which you will probably hear about more in the coming decades, and that's called precision or personalized medicine. As you know, when we develop a drug for diabetes, for example, it's developed for all diabetics, whereas all the clinicians, the doctors, they know every diabetic is different. So one drug may not fit one patient or the other. So there are a lot of differences between people that clinicians, the doctors, are already familiar with. Now, why do we do all these DNA studies in particular here? Well, that is, of course, because DNA is the blueprint of life. Once we understand how DNA is working, then we understand everything about biology. So it can help us to understand the mechanism of diseases in this respect. And that, of course, also has influences on treatment, because once you know a mechanism, you can start to fool around with it and intervene with it. And that helps you to come up with new drugs. But also, and that's an important part of this whole talk, is DNA. You are born with your DNA, you die with your DNA. It stays stable over your lifetime. So once you know such relationships between DNA variation and disease, for example, or how you respond to a drug, you can use that information very early on in your life. And of course, there's a big debate now on if we have such knowledge, when should we apply this particular knowledge? Should we do that in a hospital where it's in fact too late already because then a person is ill, or should we maybe do it in the heel prick when babies are born? Then we have a whole lifetime of opportunity to do something about it. It's a big debate about it. In any case, prediction is an important aspect of the research we do with DNA, both in terms of your risk of diseases you will develop, but also, and that's also clinically very relevant, how do you respond to treatments? Well, we are very familiar now with a very well-known aspect of COVID, which is the side effects of the vaccination. Of course, I'm totally biased. I'm a geneticist. I think there's something genetic behind these people who develop this thrombosis-like side effect for the vaccination. But it's very rare, luckily. But it is a, by so far, well-documented side effect, but very rare. But it's important to understand, are we able maybe to predict this when we have that knowledge about the DNA variation that is underlying this? This is just an illustration to you of all the work that has been done over the past 20 years or so in understanding the differences between humans in their genome, in their whole of DNA. So what you see here are all these letters, the building blocks of DNA, A, T, C, and G, and they come in a sequential order of about 3.3 billion base pairs. And these base pairs, we now know, can differ between individuals. And they can differ big time. You see here a simple one. For example, here's a T at this position, but it can also be a G. So that's called a single nucleotide polymorphism, or a SNP. We now know they're very, very frequent in a genome. we estimate currently over 350 million places in the genome out of those 3.3 billion are variable between individuals. So that's about 10% of your DNA can be variable, can differ between you and your neighbor and people you know. We also know that these variations in the DNA are occurring very frequently in the population. These are not rare events. Half of this classroom will have a certain variation at that position. The other half will have another base pair at that position. Very frequent. So, for example, you can say that if you take your neighbor, you will differ by, on average, 10 million positions in your DNA. Among those 10 million, there will be at least 20, maybe 50, which are helping to develop what we call Mendelian disease. Mostly you need 2 of these rare pathogenic mutations, but each of us carries many of these single Mendelian mutations. All these concepts are now becoming aware, we are becoming aware of those, and they are becoming more and more urgent to apply in a clinical setting. I'll come back to that at the end. So many DNA variations in the genome, but also in the population. We have developed several techniques to analyze DNA. I will not go into detail. I will share the slides with you if you want to have a closer look at it. But one of the most successful ones has what we call genome-wide association studies, or GWAS. GWAS is a very familiar technique by now. It was developed early 2000, and it basically consists of putting your DNA after some laboratory manipulations on a thing we call a chip or an array. And this array has many probes, about 1 million, which will interrogate your DNA. So the results you get back are your DNA for 1 million positions where we know there are variations between individuals, and it will give your particular base pair at that position and what we call your genotype at that position. We illustrate those, let's say, analyses of variations in what we call Manhattan plots. I will not go into detail, but what you see here are all the different chromosomes we have in different colors. And each dot is one such a DNA variation, a SNP, which has been interrogated in relation to, for example, a disease. So this SNP here, this green dot, says that this particular DNA variation has something to do with the disease for which we analyze it. Let's say COVID susceptibility, or diabetes, or Parkinson's disease. So by doing this analysis in many subjects, you really need thousands, if not millions of subjects. We've come to discover many of these DNA variations which are related to diseases in general. And we do that by combining many big data sets. So we have to collaborate with each other. Now, we all know that collaboration is very difficult between human individuals. And there's an interesting Bulgarian artist. His name is Janko Svetkov, which make these maps of the world based on prejudice. And I show you here the map of Europe as viewed from the United States. So I used to call this Europe according to Trump. But since he's no longer in office, I changed it to that Europe is blessed with many diverse cultures. But I'm sure you will recognize many of these prejudices. Like France has smelly people, their mummies in the UK. Of course, this is the Socialist Union in the Scandinavian countries. We are Sodom and Gomorrah, anything can go here, etc. Now, this is, let's say, real life, but in science, it's not so difficult. We in Rotterdam think that we do things much better than they do it in Amsterdam, or in the UK, or in Poland, etc. So you have to overcome all these differences between the countries. And on top of that, of course, our politicians make life difficult because they are erecting walls now, the Brexit wall. Maybe Germany is too big for the European Union. Turkey wants to join the European Union. Do we want this? There's Catalonia wants to go separate, and Ireland maybe wants to join again, and etc. We have all these walls. So life is not becoming more easy with all these politics going around, but in science, we've shown how it can work, in fact. And this is where I come to COVID, because early in 2020, when the outbreak was happening, we as geneticists around the globe started to collaborate with each other. And this was not new because we were doing this already for many other diseases, but we decided let's do it for COVID. And what you see here on this map are all the institutes and research groups, including ourselves in Rotterdam at Erasmus, who collaborated with this large effort to understand more about the genetics underlying these differences in how people respond to COVID. Who are infected, how is the disease progressing, and who is responding differently to these treatments. Again, I won't go into the details, but it suffices to understand that there are over 2 million subjects in this analysis now. That includes cases, so COVID cases who are hospitalized, but also controls. You need to compare a case to a control always to understand what is specific for a case. And what we found now is 17 of those SNPs I just illustrated to you. The call I had before this lecture was about this consortium, so I can give you the latest news now. We have 17 hits, and this manuscript is currently now under review with a scientific journal. We have helped in that respect to allow much more genetic analysis to be done. That's our role as Erasmus MC, but we're also contributing data to this whole effort. You can see that back here in this overview of all the data. So this just illustrates to you that looking at COVID susceptibility is just one of many aspects of human disease, human behavior, appearance, etc., that we are analyzing by this approach, by collaborating with many people with this particular genetic analysis technology. This shows you a very brief overview of what this has all resulted in. Again, the details are, the devil is in the details, because you can see here the number of subjects that we have analyzed with all these techniques for all these different aspects and diseases. For example, in breast cancer, we've analyzed 170,000 people. But here for blood pressure, we're now over 2 million people we have analyzed with this approach, like for COVID genetics. The best one at the moment is height. How tall are we? That's also subject to this very large collaborative analysis. Over 5 million people, genetic data on 5 million people are included in this analysis. And now, and that's called the explained variance. That means how much do we understand of the variation in height in this respect? Well, half of that variation in the population we understand now is due to genetic factors. And this is something that we can use in a clinical setting. Not so much for height, of course, but we can use it, for example, for your lipid level, your cholesterol level, your blood pressure, breast cancer, age-related macular degeneration and eye disease, etc., etc. There are literally hundreds of diseases and characteristics which are subject to this particular genetic analysis. And they result in what we call polygenic risk scores. So each of you has a score for all this combination of all these SNPs, which tells something about your risk for a particular disease. So through applying this technique of GWAS, what have we learned in fact? Well, as I showed you with all these prejudices, it has brought about a different culture of doing scientific research together in a collaborative setting, by harmonizing data sets, by working together and replicating each other's results. Very important in science. We've also understand much more about all this complex genetics. So what is underlying the genetics of diabetes or the genetics of COVID susceptibility in this respect? And that knowledge has already led to new intervention based on this genetic knowledge. New drugs are being developed. Drug development takes a long time, of course, as you all understand, usually 5 to at least 10 years or so. We've also developed some insights, genetic tools, which we can use in a clinical setting. For example, these polygenic risk scores, because they can be assessed in each individual and they allow a risk prediction for individuals. Again, this discussion comes about now, when should we do that? Late in your life or very early in life? And last but not least, this is a very technology-driven field. We are always dependent on ways to analyze DNA. So this technology is very important in this respect, and that's where I come to my almost last slide, which is about this technique, because that has brought about so much changes in our field. And this array used to be very expensive. It cost about 1,000 euros per sample. By now, it is less than 30 euros per sample to analyze your DNA for 1 million positions. And from those 1 million genetic markers, we can deduce many, many things. So you see them here, polygenic risk scores. We can do pharmacogenetic analysis, so telling you how will you respond to drugs. We can analyze your HLA and blood group types, which are important for transfusion or organ transplantation. We can determine your ancestry, from which continent or subgroup do you come from, etc., etc. And that's why all this together now leads to people thinking about, should we use this in a clinical setting? And that's the last slides. And that's what we call the GOAL project we have ongoing here at Erasmus. So the long-term plan, we're not doing that right now, but the long-term plan is to genotype every patient that comes into the hospital. And we see on a yearly basis about 300,000 new patients. So we want to analyze their DNA before they see a doctor. That DNA is then analyzed by such an array, or maybe another technique. It is looked at by many different experts within Erasmus, from the Department of Clinical Genetics, from clinical chemistry, our group, etc., etc. That data is then being discussed by the doctor with the patient to understand more about what is the diagnosis, in fact, what treatment should we give you based on your genetic profile. And also for the longer term, once the patient, of course, leaves the hospital cured and happy, to also help him or her with a profile that can help this person to change their lifestyle, for example. For example, we might have uncovered that there's a big cholesterol level problem there, and this person didn't know about it. And you can change it very easily with drugs, or maybe just with your diet change. So this can be very helpful for patients who come in for a certain disease, but are going out of the hospital with many other advices for them. This is currently a pilot project. I want to make this very clear. We're not doing this yet, but it's really something that is developing in the future, because many groups in hospitals, but also outside hospitals, are doing that at the moment. And also outside of the hospital, think, for example, about companies. One company is called 23andMe. They offer this kind of analysis for less than $100. We think personally that's maybe not the best place to do such analysis. It's better done in a hospital setting, but you see the societal development that are ongoing and which are important to understand also when and how should we use this information. With that, I would like to leave you just to give you a little flavor of what's happening in the genetic arena, including work that's being done for COVID host genetics. So as I said, we have uncovered a dozen of these genetic factors. We're now looking into how can we apply this in the clinic, maybe by helping people to understand, yes, you're more susceptible to COVID and you should take precautions by resulting in not becoming infected. So prevention is always better than curing, of course. Of course, I stand on the shoulder of many people who've helped me with the research. I won't go through all the names here, but it's not me, it's this group who's giving you this, this lecture. So thank you for your attention.
Thank you very much, Andre. And it's wonderful to see that we can collaborate together around the globe for the human good, I would say.
Yeah.
That's definitely something also for the trust in science, perhaps. Now I'd like to go to Agnieszka. She will take us to a even more futuristic tale. Please go ahead, Agnieszka.
So do you see my slide?
Yes.
Okay, so I have a little bit a disappointment, I guess, because I will not talk about the project I've done with amazing artists, that is the Arizonauts, but I believe you have a link to watch it. So for those of you who have watched it, this will give you a sort of background of what I'm doing. For those of you who haven't yet watched it, this should explain things as well. So what I would like to talk is a little bit about my research on the notion of contamination, which basically carries the question on how to care before risks. My background is in philosophy, and somehow I quit, you can say, the hard university or academia and become a lecturer at Art Academy at Aki in Enschede, where I designed and ran a program of biomatters. And in the beginning of the pandemic, I wrote an article living with an affect as contamination, which was basically a sort of cry out on what is happening, and also in the context of my feeling guilt that I, for the last 6 years, I was busy with the notion of contamination when studying the works of bioart and biodesign in the context of philosophy. That suddenly, how could I praise for contamination while it carries, like an actual lives and can influence our bodies, in very literal ways, right? But then somehow, after reading, reminding myself what is it that I'm actually doing, and reading a book of Anna Boyer and Undying, where she wrote, "Disease is never neutral, treatment never not ideological, and mortality never without its politics." Somehow I embraced my concept of contamination and and continue to to think with it. All right, so how to live within contamination. So this is what I wrote. Contamination as a condition of our living bodies expresses the relational character that our bodies have never been pure, that we are porous and leaky bodies conditioned by the encounters with multiple human and non-human bodies, be it other humans, animals, plants, bacteria, viruses, particles, thoughts, experiences, and minerals, and more. Where I basically argue that contamination is our way of being. So what does contamination mean? So if you look at the theory, the use of the word of contamination within different disciplines, basically you have these two meanings. So it is positioned against idea of purity, clarity, or objectivity, usually used within the epistemology, within the methodologies of science, right? So within the field that are busy in gathering true, valid knowledge. And contamination is also used as a fuel of creativity through new relation and new bodies, where you think about how bodies are, how new bodies emerge. And here it is more about this ontological understanding of how change is possible. So within the history of science, laboratory practices were based on this understanding of contamination as a threat, right? And therefore the principle to control these conditions became universal. So contamination was and is juxtaposed with the sense of purity and certainty about scientific findings. And here scientific, I'm not only talking about hard science, right? I'm talking also about humanities, but also with the sense of autonomy and rationality of practicing scientific methods. So the pursuit of purity has been strengthened by the fear of losing control, losing the power and position within knowledge-making practices and their institutionalization. So this is what I wrote, right? We are trained by the commodification of risk to release ourselves from dependency on the body, liquids, objects, by putting them into the categories of fetishization, of logic of market economy. We are told to value independence and autonomy of identities, because only in the logic of firm identities can hierarchies sustain themselves. So what I argue is that actually contamination allows us to think about risk otherwise, to think about risk as not in this logic of constant commodification, of constant control. So contamination is not, of course, a neutral state, right? It implies this risk. It cannot be controlled. And here I follow in my research, the philosophy of Spinoza. And if you are interested, also Deleuze reading, he's a French philosopher, Gilles Deleuze reading Spinoza, where this understanding is that each encounter, with bodies, right, with different living bodies, but not only living, depending on how you define life. Each encounter may change you, transform you. You may flourish when meeting another body, you may mutate or die. Living within contamination is living within the omnipresence and inescapability of uncertainty. It is not a state of choice or control, but care. So contamination in that sense, I argue that it's a constant attention and responsibility before each encounter. It is because in the moment of encounter, bodies may change, right? You do not know what bodies can do. Living within contamination is living with this risk of care. And it is in that sense not about caring for safety, caring for profit, but risking to care before another body. So how to practice contamination? In a way, I already started this before the pandemic. Somehow we have time before and after, right? But more and more, it becomes more materialized also in the context of what kind of question are we asking ourselves today. So in my practice as an educator, as a researcher, who is trying to change and ask this question of contamination within educational practice in a precarity. I elaborated this program Aki Biomatters, which is it started as a research on biomaterials within philosophical problems, ecological problems, political problems, ethical problems, life science problems, using all these questions and methodologies. But in that sense, risking the autonomy and clarity of methodologies for the sake of searching questions, not answers. Practicing to think with the value of importance rather than truth. So in a way, like this urgent question that I'm now busy with and researching, and in a way the video of Aerosolnat and the whole research of Aerosolnat is about re-enchantment. So how can we re-enchant the world through re-enchantment of methodologies, knowledges, and institutions that perform as gatekeepers of truth? And for instance, I follow here, if you're interested, the work of Silvia Federici or Achille Membe, who writes, he's using the word pluriversity, which means practicing knowledges produced through epistemic diversity. So in Aki, in the academy, you see now some images of the space we build up, where the students have access to different tools, that are usually only accessible to students in life sciences. So using microscope, building your own tools through biohacking recipes, in order to being able to to ask those questions using the tools that you might not feel comfortable, that you need to get yourself dirty as an artist and designer in order to learn how to be accountable, right? So questions, so it's about asking questions, not in order to solve them, again, to control them, but rather to learn how to care, be accountable and responsible before them. Yes. So this is just a list of some of the questions within the program that we are raising. So I will just go through them. So whether we can imagine and pursue life forms, practices, habits, and desires that will change rather than facilitate our lives. How can we change the anthropocentric gaze? How to think about what is defined as living and natural? What are the margins where we can care fully, create with life, and what are the implications of life manipulations? How to practice living and dying bodies in art and design, and how to live with contamination and its risks. So in that sense, the practice of contaminations, right? It's about creating the conditions where these questions can be posed and be researched across disciplines, but again, not in order to control them, but to live with the risk that they imply. And if you are interested, to actually see, because of course this is very short, this is like a glimpse, a provocation, you can say. If you are interested, also the students made a publication of their work after the first wave of the pandemic, where they couldn't work in the biolab anymore. They set up their own biolabs in their own home kitchen. And the image you see, actually, it was the last classes we had just in the same day that the Prime Minister announced that they go into the lockdown. And that's it. So thank you so much. If you have any question, I would be happy to answer.
Well, thank you very much, Agnieszka. That was quite food for thought from how to practice contamination and search for ways of re-enchantment and to find new ways of and forms of living. I'd like to open the floor for questions.
Hello, I'm Emily. I will be representing some of the questions that we have prepared and that have come along. I would first like to start by saying thank you for presenting to us. And then my first question is for Luc. You were talking about kind of like a shift in the paradigm that social factors are now also being included in models that usually focus more on technical factors. And we were wondering if your models also include, have like a holistic approach in the sense of now you are predicting models for COVID, but are also the effects of the measurements on mental health, for example, included to a certain extent?
Yeah, that's a very relevant question. It is, you're really touching upon the challenge of this particular pandemic, but really any pandemic, that it is such a multidisciplinary problem. There's not one single discipline that can answer it all. As an infectious disease modeler, my expertise is in the technical part, of course, and this aspect of the behavior and beliefs. I really need to collaborate with humanities and social scientists, for instance, medical anthropologists and psychologists. But I am not collaborating with people who are actually working on mental health, and I imagine that that is kind of a different field that you would not necessarily need a model for of my type, but you really need to go out there and talk to the people, I imagine. So I recognize the importance of your question. I'm not sure how I, as a person, can contribute to it, except for showing what, clearly communicating what a pandemic means, or what an infectious disease means, and how it might affect our behaviors, and getting that conversation going. That's the best that I can do, I think, to contribute to the holism that you are referring to.
Yes. And if we're talking about the future and about predictions, on a personal level, is there anything that you feel like needs to be the next breakthrough? Like now, for example, social factors are being integrated. Is there something that you feel like, hey, we also need to start integrate this?
Well, this social stuff is really really new in the sense that it has been considered in the past, but like in economics has done for decades, this human aspect of transmission of infectious diseases was considered to be rational. Like economics considers human behavior, for a long time considered human behavior to be rational in the sense that we're always trying to maximize utility, which is not true. And likewise, humans do not behave rationally about decisions with regard to their health. We have preferences, we have habits, we have social pressure. And this is really the next thing that needs to be incorporated in infectious disease modeling as well. So that, I think, is the next move.
And then that makes me think, touch upon the role of communication, maybe. Is there, because when you talk about making models that predict, see humans as factors, as aspects, but even though the social factors are included, then it still needs to be translated, right, for those models or for things to happen in practice. Do you think communication plays a big role in that?
Absolutely. Even within the scientific field, there is a lot of fear and misunderstanding about models as being either something predicting the truth or either that is totally scary and unreliable. But on the other hand, we're so used to hearing the weather report and we're totally fine if it doesn't rain tomorrow, even though they did say so. Yeah. So there is definitely a communication challenge there. And it's also about public education, I think.
Okay. Yeah. Okay, clear. Thank you so much. Then I have some questions for André. Yes. I heard you talk about, touch upon the topic of diversity, and was wondering of the importance of diversity in science, and if you have experience, for example, collaborate with colleagues that have a different background, let's say in the field of science, like female representation, but also colleagues with a different cultural background, how important that is within science.
Yeah, in fact, at two levels, because we collaborate with many colleagues around the world, as you have seen, in all these different sorts, yeah. And that includes many women, many of unknown gender, many of different ethnic backgrounds. So that's a very diverse group. But also content-wise, in terms of that we are studying genetic diversity. So as you might know, most of the data that we have collected, I think as scientists in general, comes from white, Caucasian, rich, wealthy, industrialized countries. And this is, of course, not how the world is. So there's a big movement in our field, in genetics, but in biology in general, to much more look at this ethnic diversity, as if I may call it. So use the genetic diversity between individuals from the different parts of the globe to understand much more about the underlying biology of diseases, characteristics, etc. Of course, there are challenges there because it's not so easy, for example, to get African samples which are well documented, well stocked, and coming from, let's say, high quality, great epidemiological studies, simply because the whole infrastructure is not there. So you have to sort of colonize such areas, and that is what's happening by all different kinds of scientists who go to these areas and, in collaboration with local people, develop such an infrastructure and create data there. But that has only been ongoing since, well, on a bigger scale, since 5 or 10 years now. So we have a big gap there to cover. But we're making explicit use of the ethnic diversity there.
Yeah. So you feel like a lot of dogmas are being broken right now? Well,
as I said, there's a real culture change in doing scientific research because of this collaboration. And for example, I'm trying within the funding organizations in the Netherlands to introduce that culture change, because many of the mechanisms we have to give funding to people and to grade them in terms of quality is based on this individualistic, me being the first character of science. And I think that's totally wrong. Science should be for the good of mankind and not for one scientist having the glory of winning a Nobel Prize.
And
you know,
when
that's
you're
nice, but...
And we are talking about proposing this within the Netherlands. How is that being received?
With different opinions, as you can imagine. And because it will take time, of course. We cannot suddenly change this whole culture. We have to do this gradually. But the starting point, in fact, is replication. Yeah. Replication of observations requires you to collaborate with, let's say, your biggest competitor, yet in order to get a similar answer. And that is also important. You were just discussing communication. That's also important for the outside world looking at us. Mm-hmm. Sometimes, if you've seen it with COVID also, they ask one scientist in any discussion program on television who will give his or her opinion. The next scientist will give a slightly different opinion. People get confused. Yeah. And that's not good for the image of science. And that's why I think we should get our act together and come up with a much more uniform, let's say, communication to the outside world about this. Yeah. This will help us really.
Yeah, and I can imagine that also what Luc was saying, that education is also key in this.
Of course, and that will take generations. Like within Erasmus, we have education programs for doctors. Yeah. And you see a big difference between the young doctors, which are educated now much more in this open culture of collaboration and using this all kinds of different disciplines, compared to the older generation doctors who come from a totally different viewpoint on quality. What should I believe? How should I respond? How should I act? But it also is within the whole ecosystem of our healthcare, because that is all based on, you know, where's the money coming from? What should I do to get more money?
Yeah. And is there also, is the side question, when what people now, generations, do you also have an influence if you feel like maybe from experience now that the curriculum maybe needs to change or needs to be adapted? Is there room for that kind of input?
Oh, sure. But that's always work to do. For example, in Erasmus, we have different education programs in the field of genetics given by different groups, and that's not coordinated. So I would very much like to see that much better coordinated. Just taking genetics as an example. Yeah. And that's how a lot of the things are siloed in our organization, but also in society. For example, if a patient walks into the hospital with diabetes, it's a problem if you discover that that person has cancer. Yeah, underlying cancer, because that's a different department, different DBC, trouble. Yeah. Whereas it should be our responsibility to take the holistic view of that patient and say, you know, whatever you have, we'll cure it.
Yeah. And well, maybe we can use art to speculate new structures.
Of course, I was very inspired by Agnieszka's talk about contamination, because one of my other hobbies is microbiome, which are all the little bugs in and on our body, and COVID is one of them. Yeah. And as we hopefully all now realize, we are continuously spreading all kinds of bugs to each other. Yeah, continuously. Yeah. I do not regard myself as a human. I see myself as a zoo. A walking zoo with all kinds of bugs in and on me, and I'm exchanging them with my environment. Most of the time this is okay, because they don't hurt you, they might even make you stronger. Sometimes it's a bad bug, like COVID, and we should take precautions for that. Yeah.
Thank you very much, André.
One remark for Luc, you say the weather forecast doesn't bother us. Not if you're a farmer or a wine culture, yeah? You care very much about the accuracy of the weather prediction.
Yeah.
So it depends on your audience. Yeah.
Okay, we would like to ask some questions to Agnieszka as well. We were thinking about the impact that art has had on the medical field, and we were touching topics such as controversial topics as abortion. What do you think how do you think art plays a role in the medical field, that maybe creating attention, bringing awareness through art as in such a topic of abortion has impacted the medical field? What do you think the role can play? What do you think art can play the role of in this field of the medical world?
Yeah, so I'm not an artist, right? So just to make it clear, right? So I also analyze and look at art, and I play a little bit with it. But to be honest, you know, and I'm not a scientist, so I don't know how actually, you know, the medical scene, you know, takes to heart at the, you know, the art, right? And usually from, you know, from the perspective as the one who was researching it, usually the scientists look at art and see it as a PR for the research. Of course, there are exceptions, so not to diminish any, you know, collaborations, but the impact, I would say, like of art, or the ideal scenario for art would be to, you know, challenge the existing presupposition, to actually not provide this unified image of what, you know, science can do, what health is, what is what is life, right? So unlike, you know, because Andrea was just saying, like, it would make problems, the problems would disappear if we would have this unified version. But I would say the problem is that we are still striving for this unified version, this unified answer, as if this would then help us in something, right? But life is not universal. Like you just said, you know, we are multiplicities. Our DNA is not one, is multiple, right? It's quite amazing that you are looking at microbiome, because exactly microbiome, the whole field of epigenetics, questioned and totally dismantled, I would say, right? Like the presupposition of the genetic determinism.
Oh yeah. Well,
And
coming to your remark about DNA, an important observation is that there is not one good genome and all the others are bad. You know, they're different.
Exactly, yeah.
And we have to understand what makes these differences in order, in our case, in the hospital, to help cure diseases. But the realization is there's not one superior, good, natural, normal genome. We're all abnormal. That's the take-home message.
Yeah, and that's quite amazing, right? But yet we, you know, I'm talking about the global north, right? The Western culture. We somehow we are trained to have, you know, to be able and have tools only to talk about the certainty, right? So in the moment, going back to your question, when art suddenly, you know, challenges you, provides you this multiplicity of perspective, right? In the context of abortion, which is quite you know, like a close topic to me because I'm from Poland, and I don't know if you know the what is happening over there. But you know, for me, you know, this is not a matter of philosophy. This is a matter of human rights. But in the context of, you know, how we understand what is health and how we understand, you know, life and bodies here, you know, there is a room, so much room for complexity and for challenging and for learning how to live with the uncertainty that art can do. Yeah.
In fact, you touch upon a very basic thing in biology. There's evolution, eh? So evolution very much profits from diversity and from interaction between, let's say, environmental sources and, in my case, then the genetic code that is propagated through generations. That is all driven by this interaction and diversity and evolution. Yeah.
So as I understand, Agnieszka, from your presentation, microbiology plays a big role in your life, and you are also implementing philosophy to microbiology to also understand the world. So you take something very small and apply that microscopy to something very big. Most people here probably have not gone into depth of microbiology. So is there something that you think microbiology has taught you that you wish the whole world knew?
Yeah, contamination.
Contamination.
So I don't know, for those of you who would like to know more, once the museum are open, I strongly recommend to go to Micropia. It's the first in the world museum devoted to microbes in Amsterdam. Where, you know, there is this knowledge of, you know, non-pathogenic relationship with microorganism, and what is more like conditioning, right, relationship. And, you know, like within my classes with artists and designers, when I look at the students, right, who never thought about it and never heard about microbiome, and when I teach them how to grow, you know, their own microbes, right, when they can see their own microbes through their own eyes, something is changing, and indeed they start to in question this belief in the autonomy of their own bodies. They believe in a sort of control, that you should control. And of course, I'm not, you know, I'm also like cautious, right? Because like I said, contamination is not, you know, a happy state. It's a risky state, right? But it doesn't mean that what we like, the mindset of today is to immediately disavow from any responsibility, quickly control and, you know, contain any possibility of change and difference. And this is something that you know, we are now in urgent need to learn how to think and practice about.
Mm-hmm. And to learn how to manipulate it in a way, because we are manipulating the status. Like you were saying, if the majority of the people are against abortion, it doesn't become, it's not about care anymore. It's about manipulating in such a way that it becomes a right.
Well, in the context of abortion, I have very strong, like, I have political, you know, this is a woman's right, and there is no debate. There shouldn't be no debate about it, right? This is part of human rights. Yes, I was talking.
Yeah, no, but it's because I sorry I took it as an example, but it's because when you said not a state of choice or control, but care, I thought that was a really strong political standpoint as well, and I like it. But,
we do not know also how to care. Like, very simply, we do not know how to care. We have a concept of taking care of. And the notion of taking care of is very close to control, right? So you want to taking care of from, if you take care of, you have a position of power, right? And caring, and I used the word caring before, it's also from Gilles Deleuze, he's a philosopher, where you acknowledge the agency of the other, and then suddenly you have to negotiate, you have to take a risk, because you don't know what will happen. And this risk of care is something that we need to learn.
Okay, thank you all of you for answering the questions and also engaging with each other.
Thank you so much for wonderful questions.
Yes.
Thank you. This was a lot of fun.
Thanks. Yeah.
Yeah, thank you, everyone. So this was the end of this series. I think we learned a lot. I think this time was also, it's quite an interesting way to finish with this, on collaboration, diversity, contamination, not being afraid of being how do you say that? A common, that you're contagious, and that you will contaminate others with your ideas, with your care, with all kind of stuff. I think risk is also very interesting. Everything becoming more personalized means that everything will also be more uncertain. because everything depends on so many different reasons. So we really have to collaborate and listen and take care of each other and of ourselves. I'd like to thank all of our speakers today, Luc and André and Agnieszka, of their very, very inspirational insights. And I'd like to also, of course, also as a part of the Science Gallery, I'd like to thank Hill and Marina very, very much for being able to organize this wonderful collaboration, and also, I hope, inspirational meetups of people and students and artists, scientists, and everyone, and that everyone will have a more broader view of where we have to go to. We are, as the Science Gallery, we're really looking forward to what you are, as students are going to show us in ways of ideas, processes, and what that really will bring us at the end in May, I think. We will take all your ideas and processes and products, and we will take them as for our the basis for our exhibition and educational program that will be opened on the 24th of September in the Night of the Researcher. Yes, so I hope to see you all there, and I hope to see you also in May, where we will be able to see what comes out of all the researches. I'd also like to thank Inger for all her help and very well making sure that everything also works very well with all these kind of Zoom meetings, which is not always as easy as it looks like. Well, thank you very much. Good luck. Have a lot of fun. I think you have enormous amount of inspirational information, and see you in May.
Before we leave, thank you also. Thank you, Fred and Inger. And of course, you have so many people already. But of course, on behalf of Marina and I, thank you all, and did a tremendous job by helping to organize this kickoff. It was also your idea to have this 4 days intensive lectures, and it was really intensive. It was quite inspiring too. And thank you again for organizing this, Inger, for this well-performed and well-working thing in Zoom, Wiegert for your moderating, and also Fred for your moderating role in this. And of course, all the presenters, researchers and scientists, thank you again for your inspiring and also very clear communication in language, because it also has to be understandable and accessible for the students. It was also something that we asked for, and it was well done, and I think everything was very clear. And of course, students, thank you. I'm very proud of you. You did a very good job. Beautiful questions. Yes. And but I'm also very curious what will be the now end result. I hope the processes are more important. Just as Agnieszka said, it's more in the process. And now it will be shown in May, and hope to see you all again and in the presentations. So thank you, and enjoy the weekend. So it will be hopefully a little bit better weather, but we wait and see in the weather forecast.
Yes.
Okay, guys, thank you. Enjoy your days.
Thank you. Bye-bye.
Bye-bye. Thank you very much.
Thank you very much.
Thank you. Bye.
Bye.
Bye-bye.
No.
Oh, Marina, you were still in. Afterwards, you were not in anymore.
Ja, nou dat was echt een